My talk will be about reproducibility in the life sciences. Reproducibility is an issue that has been quintessential for the scientific method since the very beginning of scientific investigation in the Western world. However, there's new issues and new trends that are continuously changing, especially in the current era where reproducibility is taking on new challenges with the urgency of information that needs to be disseminated. Most research practices in most scientific fields have the challenge of working with limited data. Small data sets means that solo, silent investigators who are struggling to make new discoveries in small teams are using suboptimal sample sizes, less what they would probably wish to have under ideal circumstances, to try to find new things and validate them. This means that unavoidably there will be a lot of cherry picking of a few results that seem to be the most promising. Much of that research is post hoc. Usually statistical inference tools are very lenient with frequency values of p less than 0.05 being considered enough in many fields. No registration is happening ahead of time for the research being done, no data sharing and no replication. This sounds like a nightmare, but actually in most fields this is probably the most dominant paradigm, and even though many disciplines like genetics have made great progress to overcome many of these limitations, this is a challenge that we see happening very frequently in most fields and applications. For example, this is an empirical evaluation that I did with Dan and Sukes from Cambridge, where we looked at a large number of papers published in neuroscience, psychology, and medical journals. The common denominator was that if you looked at the effect sizes and the degrees of freedom, which is practically sample size of these studies, most of these studies were vastly underpowered to detect what might be meaningful effects. Concurrently, we have a new paradigm that is becoming more common in some fields, and I think that in many genetics and genomics fields this is already happening in large scale. We have big data. We have sometimes the opposite problem. Instead of having very small sample sizes, we have extremely large sample sizes. This means that we have overpowered studies that are likely to find significant results no matter what. Many of these data sets have been compiled without any particular research effort or research plan or protocol in mind. For example, they may be electronic health records or other pieces of evidence that are accumulated routinely. There's a lot of cherry picking also in these data sets. There's a lot of post hoc research being done. Sometimes statistical inference tools are more sophisticated compared to the statistical tools that are employed with the typical stereotype of small sample size research, but many of these inference tools tend to be idiosyncratic and without consensus across multiple fields. Most of that research is also not registered, and even though given the type of data being involved, there's usually more data sharing, very often that data sharing is inefficient, not very effective, not very transparent. The data sets are not easy to use. The data codes are not such that would be easy to understand by different investigators. In the current era, since the advent of the COVID pandemic, we have some added challenges that have emerged. There's clearly perceived urgency because we're dealing with a major problem that we need to find urgent solutions. And this means that lots of scientists are working with very high speed, but at the same time probably without paying too much attention to issues of reproducibility. In many of the questions that are pivotal to answer in COVID research, there's sometimes very strong groupthink or beliefs that are entrenched and they're very difficult to challenge. And this means that reproducible research becomes even more necessary to be able to compare notes and to arrive at safe conclusions. There's wide use of press releases before any paper or preprint is released, which means that a lot of information is disseminated with the minimum amount of ability to do any reproducibility check, let alone understand what is the machinery of the research being done. At the same time, we see increasing use of preprints, but many people are not familiar with preprints. They do not understand what preprints stand for, how they should be used, how they should be interpreted. Sometimes they see them as immutable final products of research, while in fact they're complete research or research in the making that is being released for crowdsourcing of review comments and contributions by the wider scientific community. This results to a situation where we have lots of people who are not scientists I would argue in a charged environment, they may have even some sort of mob mentality, or even scientists turned into mobs instead of the typical type of scientific debate and improvement of the literature that you would like to see in a rapidly evolving field. In the last several years, several fields have tried to do some soul searching and to challenge the notion that their results are reproducible. One of the most classic examples is that paper published in Science 5 years ago, where a very large number of psychologists look at some creme de la creme papers published in some creme de la creme journals of their field. About 100 papers they tried to reproduce, and depending on how you read the results, the exact numbers may be a bit different, but roughly 2 out of 3 of these papers could not be reproduced. The p-values on the left-hand panel and the effect sizes on the right-hand panel were very different in the original studies versus the replication efforts, even though the replication efforts tried to be as faithful as the originals. This is not news to the genetics and genomics community. Many of us who have worked in the field for many years would remember candidate gene studies. There were tens of thousands of them that were published in an effort to look at one or a few genes and one or a few variants in terms of association with specific phenotypes. And based on what we know now, the credibility of much of that literature was very, very limited. When we went into the genome-wide association era and we could go back and check how many of these associations could be replicated in this evaluation that I'm showing you here, only 1.2% of them could be replicated, and clearly we've made tremendous progress over the years, but most of these candidate gene studies did not really go anywhere. Many other fields have also gone through some process of soul searching. Preclinical research in particular has had a number of reproducibility attempts at some high-profile papers and clusters of papers, and the reproducibility success rate varied from 0 to 20%, again in the same range as what we have seen for candidate gene studies in genetics. Many of these papers were influential papers that had substantial consequences in terms of what would be the next steps of research. For example, there were papers about drug targets which need to be prioritized, and huge investments may need to be spent on them to try to translate them into effective treatments. All this ongoing soul searching of reproducibility has also led to what I call the reproducibility wars, which is what you see when there's so much at stake, when there's so much animosity also in trying to decide whether a particular research finding, especially a prominent one that has driven research or has led to other research and to other steps being taken, has been successfully or unsuccessfully been reproduced in a reproducibility effort. We have seen such debates happen very frequently, especially in fields where even defining what it is to reproduce a research finding can be contested. Many of those debates end up in an uninterpretable zone, and there's the added layer of debate about whether we do need a lot of exact replication, whether we need more of conceptual replication, triangulation, looking at different avenues and pieces of evidence that we try to put together. I think we need both. We need both exact replication and triangulation. I think that for important findings, exact replication is a sine qua non, because we want to make sure that we're building our next steps on something that is solid. And of course, triangulation, using different avenues of evidence and different ways of life in obtaining this evidence, is also something that could be useful. A number of solutions have been proposed on how to increase the proportion of trustworthy research findings. A number of research practices could be adapted or expanded or changed in order to make this happen. Here's a partial list. Many of those are already adopted in genetics, and hopefully they can be adopted even more widely. Others may need more work in the future. I'm just going to run down the list: large-scale collaborative research, adoption of replication culture, replication of studies, protocols, analysis, codes, datasets, raw data, and results, sharing of data, protocols, materials, software, and other tools, reproducibility checks, containment of conflicted sponsors and authors, more appropriate statistical methods, standardization of definitions and analysis, more stringent thresholds for claiming discoveries or successes, improvement of study design standards, improvements in peer review, reporting, and dissemination of research, and better training of the scientific workforce in methods and research literacy. Human genome epidemiology offers some of the most classic paradigmatic success stories in terms of how large-scale collaboration and adoption of replication culture can really transform a field. This is just one example of Manhattan plots from a large genome-wide association study with meta-analysis of multiple data from multiple teams. In that case, this is a study on identifying variants that are associated with smoking behavior. Before we started doing this type of collaborative work with replication being a sine qua non, we had published hundreds of papers that suggested several hundreds of genes were associated with smoking behavior. When we did that large-scale collaborative genome-wide association testing, hardly any of those survived, but a new cluster of new genes arose that have very strong and reproducible statistical support. Registration is also another idea that has been gaining ground over time, and of course one should never argue that all studies need to be registered. There's a lot of research that is exploratory, and I would argue that for this type of research it is perfectly fine. Actually, it is the only solution that it should not be registered. It would be erroneous and even misleading to claim that research that is exploratory and has not been pre-thought in its details was actually pre-thought in its details. It gives a false sense of reassurance. Beyond that, which would be a very large share of the scientific literature in many fields, and justifiably so, you can think of registration at different levels. You can think of registration of data sets, in which case you know what that data set includes, what kind of variables, how these are defined, how rich the data are, how trustworthy the different variables are, how they have been collected, how they can be used in different ways to try to understand different phenomena or to make discoveries. Level 2 registration would be registration of a protocol, which is something that has become more common in fields like clinical trials, but unfortunately it has not been very common in other fields. Level 3 is registration of an analysis plan, which is separate than the protocol. A protocol most of the time does not include a detailed enough analysis plan, and this can make a difference. Level 4 is registration of both an analysis plan and raw data, which means that someone could use the analysis plan and the raw data, and hopefully also the code, to be able to rerun analysis, revitalize the analysis, and also use it and use these data for other purposes. And level 5 is open live streaming, where someone is sharing with the rest of the scientific community at a stepwise process the experiments to be done, getting feedback on them, revising them, running the experiments, providing new results, and then getting new feedback to design the next round of experiments and keep that going. Different fields are at different stages in terms of how much consensus there is in the field in terms of how extensive sharing can happen within their discipline. I would argue that genetics and genomics are at the forefront because probably they're sharing more data compared to most other fields in life sciences and beyond. But even in genomics, there's still plenty of room for improvement and for making the unrestricted use of public genomic data the norm. This is a paper that I wrote along with many other colleagues about a year ago, where we argued that this should be the norm and publication interests should not limit access to public data. Actually, we argued that in most situations when a data producer is coming up with some new collection of data, it is to his or her benefit to share immediately that information with the rest of the community, because it's the cumulative data sets, the cumulative data that are likely to give far more reliable information compared to each one of us trying to analyze small snippets or small fragments of data sets that may not be very informative. How about important data that have been collected in the past? Can we somehow retrieve them and then make use of them? This is what we tried to do with Tom Hardwick in a project that we call the Data Arc. We looked at the most highly cited papers of the last decade published in psychiatry and psychology. We communicated with the investigators and we asked them to send us their data, offering to curate them and make them available without requiring any funding or any support. We would do that for free, the curation and dissemination of the data sets. Very few data sets were already available, and we could manage to get a few more, but the vast majority of these data sets, even though they were not very old, could not be retrieved. What were the reasons for that non-retrieval? One might argue that maybe researchers were still interested to use the data to get credit from publications that would depend on these data rather than giving this data away to their competitors. But actually that was the least common reason. Also, one would have thought that lack of resources might have been an issue, which of course is a concern because proper data sharing does require proper resources. However, these were not the most common reasons. The most common reason was that the data were outside of the researcher's control, meaning that the principal investigators did not really have access to the data. They could not share them. Very often these data belonged to other stakeholders like companies or industry, and they had to go through a very different process in order to retrieve them. Of course, there were also legal and ethical concerns for data that had been gathered under different informed consent practices. Sometimes the investigators were also preparing their own sharing system, and sometimes the data no longer existed. Even for data that were less than 10 years old when we did that exercise, it was not uncommon that they would have disappeared in the interim. Well, we do have a very widespread interest in improving sharing and improving transparency and making more data and methods and protocols available. At the same time, there's a countercurrent. The countercurrent believes that the scientific method that has long believed in replication and sharing and communality really belongs to the past. The feeling is that now that we have a lot of data, the data somehow will just get it right. We have a lot of data and we will be able to get the right answer no matter what. We don't need to think a lot about little methodologic details. To go a step further, there's also a very strong countercurrent where many startups, for example, believe that you just need to make something work. It doesn't matter if it's a black box. It doesn't matter if outsiders cannot reproduce it. But if it works, and this is what matters. And the classic example was Theranos, that made proud statements about its approach to secrecy, to stealth research, not communicating how they do things, but promising that they could disrupt the entire healthcare with their new technology. I was the first to write a paper in JAMA where I said that Theranos has no evidence, and actually what they propose may be completely infeasible, and then their valuation may not be worth $9 billion, but just $9, depending on how much they can defend their claims. About a year later, Wall Street Journal started publishing their series of investigative articles that showed that Theranos actually was just a bubble. And very quickly, as you know, the company disappeared in thin air. One might argue that Theranos is just an exception. It's just one bad apple, and this is probably the dominant narrative, that you have a crazy psychopath who builds a company that is a fraud, and that's a major scandal, as opposed to pretty much all the other companies. And I think that this is probably the wrong narrative. I would not talk about psychopaths. I would not talk about fraud. I would just talk about the urge to find something that is disruptive and that can also get a lot of money, and cutting corners in terms of the quality of research and the communication of transparent, reproducible evidence. About a year ago, we did another survey where we looked at all the healthcare unicorns, and we realized that many of them have a profile that is very similar to Theranos. Many of them publish very little or nothing in the scientific literature, especially in high-profile scientific literature. Therefore, their claims are not very easy to appraise in terms of their validity. We repeated a similar exercise along with many colleagues looking at immunology and complement therapeutics, and we realized that there exists a very similar pattern. There's a lot of startups, but few of them have published substantially in the scientific literature. Actually, the ones that had the highest valuation tended to be the ones that had the least strong publication record in the scientific literature. This is something that is becoming very prominent in the COVID era. There's a rush, and lots of people and companies are trying to develop vaccines for COVID-19. However, there's very little that has been published in terms of these vaccines. We should not forget that for 50 years or more that we know about coronaviruses, we have not been able to develop a vaccine. And some companies that actually make the strongest claims and claim that they may be able to have a vaccine by the end of the year are among the ones that have the least strong publication record. How do reproducible research practices look across the board if we were to examine the entire biomedical literature? This is a random sample of the biomedical literature from 2000 to 2014 that we examined a few years ago, and we realized that there was very little action in terms of reproducible research practices. We found hardly any papers that were sharing data. As you realize, the few genetic papers are just a very tiny proportion in that sea of evidence and publications. We found practically nothing in terms of particle sharing. We found almost nothing in terms of code sharing. And there was little action in terms of statements of conflicts of interest and disclosures of funding, with some improvement nevertheless over time, over the years. We repeated that exercise looking at papers published from 2015 to 2017, and we did see some improvement clearly, especially for data sharing. Data sharing seems to be picking up, and actually now we have looked at a much larger sample of all PubMed Central papers, 2.7 million papers, where we realized that in the last few years there's clearly an improvement in the number of papers that have been making available data statements for data sharing. There's far less action in terms of code sharing yet, and there's clear improvements in the disclosures of funding and disclosures of conflicts of interest. Most papers continue to claim that they have something novel to say. Very few papers are about replication, and very few papers acknowledge that replication is such paramount importance for the credibility of the results in the scientific literature at large. These are the data for the trajectory of data sharing over the years from 2000 to 2017. You see that there's very little action until 2010, and then some continuous improvement reaching to levels close to 20%. We have validated that looking at all 2.7 million papers in PubMed Central. We have about 15% current sharing of data in the biomedical literature, which is not perfect, but I think it's much better than what we used to have. Of course, sharing does not mean that these data would necessarily be usable and or that these data are actually used constructively and effectively. I think we need to invest more in trying to make data sharing have more value. For data sharing to have more value, it needs to be combined with sharing of code, with sharing of particles, with sharing of instructions. And this creates a very different universe of computational methods reproducibility. When we wrote that paper 4 years ago in Science, I think that this was still in early days. There were some fields that were very cognizant about how important that is and how indispensable to really put any credibility in any scientific findings. I think the number of fields that have learned that lesson is growing steadily. Genetics and genomics have some of these disciplines that are at the forefront, but clearly there's a lot that can still be done in terms of improving the reproducibility of computational methods. How do we study the impact of changing reproducibility practices? This is not easy because it's not easy to create randomized controlled trials where individuals or institutions or whole countries are randomized to adopt or not adopt one single change in their research practices. Nevertheless, we can model different aspects of reproducibility, and we can see what would happen in different environments and settings if we try to change the way that science is done. So here's one such model that one could present. Along with David Grimes, we created a small universe of science based on 11 equations. This is a universe that includes 3 types of scientists. It includes diligent scientists. It includes scientists who may be using sloppy methods or questionable research practices, and it includes some scientists who are fraudulent. Fraud obviously is very frequently in the news, and it makes big news when it hits publicity. However, I think it's extremely uncommon in science. Science is one of the fields that has the least chance of someone being fraudulent compared to almost any other human occupation. Most scientists, I think, are either diligent and they try to do their best, or they may not be very knowledgeable and may be cutting corners and may be following questionable research practices in some of their endeavors. This is what happens, though, when you run that model. Even though you start with most of the scientists being diligent and a smaller proportion being careless and very, very few being unethical, if you reward scientists based on their achievements and based on what they have managed to produce, and you do not give differential rewards to those who are diligent versus those who are careless versus those who are unethical, eventually the careless and the unethical will take over. Why is that? You don't need 11 equations to describe this. Practically, it means that if someone can get to the same target and get the same reward doing less work by cutting corners or doing no work at all by being fraudulent, then that person will have a survival advantage in that competition. And obviously, if you let that happen, then the trustworthiness of science will decrease over time as more scientists who are not diligent are dominating the field, and they're also creating progeny of trainees who follow the same standards. What this means is that we need to re-engineer our reward system if we want to get things right. If we want to improve the quality, the efficiency, and the rigor and the trustworthiness of research, we need to pay attention not just to productivity, but also to quality of the scientific work, to reproducibility of the scientific work, to how much sharing of data and other resources is happening, and also to what is the translational impact of research. If you run through that acronym, it sounds like an electrocardiogram, PQRST. We're putting a lot of emphasis on productivity, and I have nothing against productivity. Being able to do more work, interesting work with more colleagues, with more collaborators, publish more papers, this is fantastic, and I don't think that we should penalize that. People have suggested that maybe we should not allow scientists to publish more than a small number of papers each year, and I think that for many fields, especially genetics, where there's a lot of collaborative research, this is not really a plausible or even reasonable suggestion. Conversely, I think we should defend productivity, but also work on the other QRST, because they can make a difference both in the efficiency and the trustworthiness of science. There are many opportunities to do that. There are opportunities at the level of hiring, promotion, and tenure of scientists. There are opportunities at the level of funding institutions and organizations of universities, of research centers, of anyone who has a relationship of reward and incentive with the scientific workforce. A couple of years ago, we hosted a workshop and wrote that paper in PLOS Biology on how we could use reproducible research practices to have pivotal roles within the setting of hiring, promoting, and tenuring scientists. We have a paper coming out in BMJ with David Moher and his team, where we have looked at about 150 universities around the world, and we realized that very few of them currently are using reproducible research practices and progressive criteria for their hiring, promotion, and tenure decisions. However, there's more sensitization in the field, and I think that we will see more of that happening in the near future. To conclude, there are many possible interventions that may improve the efficiency of research practices and the reproducibility of the evidence. We don't really know which ones are the best. There's many ideas, some of them have strong evidence, others are very speculative. I think we need to test as many of them as possible and be open-minded about keeping those that work the best, adopting them and adapting them to different fields depending on their needs, but not take it for granted that all of them will make reproducibility and efficiency and trustworthiness better. We just don't want to add to bureaucracy. We want to make science better. Reproducible science is not an outsider to science. It's part of the integral way that we do science every day. Transparency, openness, and sharing are likely to be important and to help. Details on how can be important, and these details need to be vetted out by the scientific community in each discipline. It's unlikely that one size will fit all, but scientists who work actively in a given field can tell what type of transparency, openness, and sharing will make their life better, will make their science more robust, will make them happier to make advances in scientific investigation. Finally, the landscape of reproducibility is currently changing very fast. We live in very interesting times. We have new expanded stakeholders who are active in shaping and sometimes distorting science and its impact. I think that we should welcome input, we should welcome criticism, we should welcome constructive suggestions on how to improve how we do science. At the same time, I believe that it is important to try to defend the scientific method and stick to what has made science successful over many centuries. Reproducible results, results that can stand the test of time, that can stand the testing by adversarial and even controversial testing. So interesting times moving forward, but also some improvements that are already happening. We'll see where we go. Have a wonderful day.